Assalamualaikum, my name is Nazatul Amira binti Arifin and I will be presenting my final year project entitled Recycling HPLC for the Isolation of Styryl-Lactones from the Bark-Twig of Goniothalamus, and this project was supervised by Dr. Norviki Bihud. These are all the outlines that I'm going to present for my project, and I'm going straight into the introduction first. So, let me begin by introducing you to the plant species that I've used for my study, which is Goniothalamus. Goniothalamus comes from the family of Ananaceae and the genus Goniothalamus. It is locally known as a kena and its synonym name in scientific literature is Goniothalamus umbrosus. It is found and ecologically distributed in the regions of Peninsular Malaysia, Sumatra, and also Thailand. It is traditionally used in treating typhoid fever, skin diseases, cancer, and also postpartum toning. Alright, so these are all the plant parts of Goniothalamus, which are flowers, fruits, leaf, and also bark and twigs. But specifically for my study, I focus on the bark twigs of Goniothalamus because it is rich with pharmacological properties. So, the reason why I conducted this research study because Goniothalamus is considered as a popular alternative medicine in East Coast Malaysia. But there are very limited and insufficient reports on phytochemical and pharmacological properties of Goniothalamus. Previous study utilized a conventional method such as column chromatography, which is not sustainable and is a lengthy process that causes unresolved peaks of the compounds. So, for this study, I've used recycling HPLC because it expedites the isolation process, enhances preparation efficiency, and also promotes sustainability by minimizing the solvent usage. There are two objectives that I aim to achieve by conducting this study. Firstly, to isolate and purify the chemical constituents from the dichloromethyl extracts from the bark-twig of Goniothalamus by using recycling HPLC, and secondly, to elucidate the structure of isolated compounds by using modern spectroscopy techniques such as NMR and also SEMS. In the chapter 2, I'm going to talk about the literature review of my study. Based on the previous reports, there are several phytochemical compounds isolated from Goniothalamus, which are Styryl-Lactones, alkaloids, steroids, and also terpenoids. These are all the phytochemical compounds isolated from the Goniothalamus, which are Styryl-Lactones, and we have alkaloids, and also we have steroids and terpenoids. It is reported that Goniothalamus has biological activities such as cytotoxicity or efficacy activity and also anti-inflammatory activity. The phytochemical compound called as gonithalamine was reported to show highly potent cytotoxicity against murine leukemia cell lines with ED50 values of 0.19 microgram per milliliter. The second report found that gonithalamine and creptimeridiol exhibit strong anti-inflammatory activity against PAF receptor binding with IC15 values of 19.7 micromolar and also 70.5 micromolar respectively, with over 60% of inhibitory percentage. Alright, now I'm moving on to the research methodology of my study. So the workflow for this study involves several steps where we start with the recycling HPLC profiling to determine the best solver system to isolate the compounds for the fractional of Goniothalamus. After the solver system has been determined, which is 70% of acetonitrile and 30% of water, the isolation process starts by using recycling HPLC. In order to get as much heat as possible, the isolation process and the injection were repeated for a few times. And finally, the pure isolated compounds will be elucidated by using NMR and also ICMS spectroscopies. So, the main instrument that I have used and utilized for my study is recycling HPLC because this instrument enhances the separation efficiency by incorporating the recycle valve in the HPLC system, which allow the overlap piece of the compound to recycle back into the column, which proves that this method is economical, practical, and also sustainable. Now, we are moving on to the chapter 4, where I'm going to show you the result and discussion for my study. So, this is the chromatogram of recycling HPLC, where the first cycle started after 22 minutes and the second cycle started after 2 minutes, where the peaks slowly separated into two peaks, which indicates there are two pure compounds, where we label it as the collector F9C1 and also collector F9C2. The minor compounds will undergo the recycling process, meanwhile the major compounds will stay in the system. So, this is the comparison for the data NMR. So, the bottom NMR spectrum shows the mixture of FNIC1 and FNIC2. And after the recycling process, the mixture will be separated into two pure compounds where will be identified as FNIC1 and FNIC2, which are referring to the major compounds. Now, we are going to elucidate the structure of compound F9C1. by using NMR. The proton NMR spectrum show 4 aromatic rings which resonate between 7.22 until 7.33 ppm, which indicates the presence of monosubstitute phenol moiety. There were 4 additional 1 proton signals at 4.38 ppm until 4.74 ppm, which indicative the oxygen-bearing methane protons assignable as H4 until H7, and another 2 additional 2 proton signal that are observed further downfield at 4.97 ppm and 5.48 ppm, assignable as H14 and H9. But these 2 protons were not oxygen-bearing methane proton because the carbon NMR spectrum revealed that the chemical sheets for their carbon were 100.23.7 ppm and 119.3 ppm, whereas the oxygen-bearing methane carbon usually and typically resonate between 70 until 85 ppm. So in the carbon NMR, the higher ppm value of a methane carbon or proton is typically related to the anisotropic dissiding effect of the pi electron in the double bond. So this gives us the idea of the adjacent carbon bonding. The carbon NMR spectrum revealed a total of 23 carbons with a carbonic carbon typically of a lactone moiety, which was observed at 172.6 ppm. Lactone moiety is the most common in the compounds that isolated from the Goniothalamus species. This is the NMR spectrum that help us to identify and differentiate between methylene, methyl, and also methane carbon peaks. In this experiment, methane and also methyl carbon show the positive and upward signals. Meanwhile, the methylene carbon peaks show the downward and also negative signals. Particularly, 2 methane signals were observed in the outfield region of 3.07 ppm and 2.46 ppm, which assignable to H2 and H3 position. 4 pairs of methylene carbon peaks were still resonating between 1.8 ppm until 2.7 ppm, and 2 pairs of methyl proton resonate between 1.5 until 1.6 ppm. The structural elucidation of F9C1 was done by analyzing quasi correlation among the protons and using HSQC to find which proton are directly bonded to the specific carbons. All the oxygen-bearing methane protons, which are H4 until H7, correlate with one another within the same ring. Only H4 correlates with another methane from the outfield signal, which means another additional structure starts from here in the shaded region. Both methyl protons correlate to the decided methyl proton H14 and also a methylene which was assignable as H14. Both of these correlated to each other and they are staying in the shielded region. And the further experiment on other correlation between the proton has helped us to conclude a molecular structure of F9C1. HSQC spectrum shows the correlation of a proton which directly bonded to the specific carbon. As you can see how the proton at H9 correlate with the carbon at number 9, and also the proton at H2 correlates with the carbon number 2. The final step for the structure elucidation was done by using the SCMS spectroscopy. The molecular formula of the carbon-23, hydrogen-28, oxygen-4 was established by SCMS experiment, which records the mass to charge ratio at 369.20, which confers the molecular ion peak for F9C1. Now we are going to the structure elucidation of F9C2. So these are the comparisons of the proton and the carbon NMR between the two major compounds F9C1 also F9C2, which show very similar values, but a little difference related to the configuration of the structure at the position carbon number 2 and also the carbon number 3. And the further experiment will be required to be conducted after this in order to figure out the isolated compound structures. And finally, I'm going to summarize my entire final year project presentation through a conclusion. In conclusion, recycling HPLC has successfully isolated two closely related structures with similar retention characteristics, and this isolation wouldn't be possible to conduct by using other conventional methods such as column chromatography. Both compounds are possibly new, and the structure's configuration will require the further experiment by using advanced spectroscopy methods, and the isolated pure compounds are identified as F9C1 and also F9C2. And now we are coming to the end, and these are all the list of references that I've used from my presentation slides, and I think that's all from me. Thank you.